Howdy, folks. Welcome back to Human Anatomy. I'm currently recording the second half of our reproductive unit. After that, I have to record our ecology and environmental units. Those will be posted for next week. Yeah, I think we should go and just get straight into it. So I'm gonna start a screen share. Let's go and share over to this screen. This is the wrong presentation, but that's just because when I full screen this presentation, boom. Alrighty, folks, we're going to be looking at chapter 18 in the textbook, Development Throughout Life. So, outlines. We're going to have periods of development. We're going to examine the periods of development in human life. We're going to talk about the prenatal period. We're going to talk about birth. We're going to talk about some common birth defects. We're going to talk about milk production by the mammary glands. And we're going to talk about the postnatal period. So human development is going to begin with fertilization. That's about where we left off on our last presentation. Fertilization is going to be the union of the egg and the sperm together. The prenatal period is going to be a period of development before birth, and the postnatal period is going to be a period of development after birth. It's just pre and postnatal being the key there. So the prenatal development can be divided into three different periods. So the first is from fertilization up through week 2. This is known as pre-embryonic. Then from week 3 through week 8, we end up going through the embryonic period. Then from week 9 until birth, we are in the fetal stage. Here are those three, along with the major biological events that are going to be occurring during those stages. So pre-embryonic, we have the fertilization event, cleavage, which is just when the cell starts to go and divide and become a multicellular organism, and formation and implantation of the blastocyst, beginning of formation of extra embryonic membranes and placenta. So this is going to be the anchoring to the wall of the uterine wall. This is going to go and be when we start developing our secondary and support tissues that are going to go and like feed and protect the fetus. Then we have the embryonic period. This is when we have gastrulation, which is the formation of the GI tract. And this is also about when tissues, organs, and organ systems are going to start to form. From week 9 onward, we're going to go and see a drastic increase in length and a drastic increase in weight, as well as the continued growth and specialization. So here are images from each of those stages. Here's a zygote immediately after fertilization. There's a blastocyst getting implanted into the uterine wall. There's an embryo at about 7 weeks of development. There's a fetus at about 5 months of development. So the pre-embryonic period begins with fertilization. The eggs live only 12 to 24 hours after the release, and most eggs are going to be fertilized in the oviduct. Although it is possible for it to be fertilized lower down, it is going to go and have the most success if it gets fertilized in the oviduct. Otherwise, it's most likely not going to be able to implant on the wall. This is going to happen within 12 hours of the egg's release, although it is important to note that sperm can survive for multiple days inside of the reproductive tract. So, major events of the pre-embryonic period. First, we have fertilization, when the male material fertilizes the female egg. Then we have cleavage, where the cells starting to split and divide and form multiple cells. And then we have the beginnings of extra embryonic material, so material that's not going to be part of the baby itself. So here, ovulation, secondary oocyte is released from the ovary and swept into the oviduct. Then fertilization, a single sperm manages to penetrate and fertilize. Cleavage, we start to see cellular division. Then we get here. By day 4, we have a solid ball of cells, and this is what's actually going to be anchoring to the wall of the uterus, and this is known as a morula. Then we have the blastocyst. So by day 6, the pre-embryo becomes a blastocyst, a hollow ball of cells with a fluid-filled cavity. Blastocyst has freed itself from the zona pellucida and can increase in size. So the blastocyst is going to anchor to the wall of the uterine and then start to enlarge. We do not start to get this stage is not going to go and start until we've managed to successfully leave the fallopian tube. We do not want to be in the fallopian tube. Now, fertilization. This occurs in the oviduct. So this is going to go and involve the enzymatic action of the acrosome. So the acrosome is that thin gel head that covers the sperm that we were talking about. It's going to be a cap on the end of the head of the sperm. It's going to contain enzymes that are going to be able to digest their way through the outer layers of the secondary oocyte, which is in turn going to then allow the genetic material of the two to mix. Yes. Now, the corona radiata and the zona pellucida are also going to going to be the two zones that are going to be digested through. So sperm enters the when the sperm enters the cytoplasm of the secondary oocyte, the zona pellucida, which the sperm has just pushed its way through, it's going to go and harden. It's going to do this by releasing different granules, basically scarification of the outer surface of the cell. And this is going to prevent entry by additional sperms. Once this has happened, they can no longer burrow their way into the egg. Now, the secondary oocyte completes the second mitotic division and is now considered an ovum. This is done through the sperm and the ovum fusing together to go and merge their genetic material. This is known as karyogamy, and it is the basis of all sexual reproduction. Now that the egg has been fertilized and it has the full amount of DNA that it's going to need to go and grow into an organism, we are going to refer to it as the zygote, the fertilized ovum. So that's going to contain genetic material from both the mother and the father. You're going to have 23 chromosomes, one copy of each chromosome in the human genome, and you're going to receive one copy from each parent for a total of 46 paired chromosomes. So this is the actual process by which the sperm is going to go and be anchoring itself into the egg. Secretions from the female reproductive tract alter the surface of the plasma membrane of the sperm. So this is going to go and start signaling the next stage of behavior for the sperm's life. The sperm contains the corona radiata, and enzymes spill out of the acrosome. Sperm contacts the corona radiata, and then enzymes are going to go and spill out of the acrosome. Enzymes are then going to disrupt the corona radiata and then the zona pellucida so that the sperm can now finally make physical contact with the plasma membrane of the secondary oocyte. The sperm is going to enter the cytoplasm of the secondary oocyte, and then granules in the plasma membrane of the secondary oocyte are going to release their enzyme, causing the overlying zona pellucida to harden. So this is going to go in what's actually going to prevent further penetration by more sperm. At this point, the nucleus of the sperm and the nucleus of the egg are going to fuse through karyoptosis, and fertilization is now going to be considered complete. Here are the cortical granules, and this is inducing scarification. Now, just realized I had my mouse on the wrong screen, so you guys didn't see me, but it's numbered and in order, so I imagine you can follow along. Now, cleavage. So, a rapid series of mitotic division occurs as the zygote moves down the oviduct towards the uterus. So that's just going to be our cellular replication. This is going to be mitosis, non-sexual cellular replication. Once enough cells have formed around day 4, we're going to have a morula, which is a pre-embryonic solid ball of cells, anywhere from 12 to like 30 or 40. Now, at this point, there is a chance that the ball of cells can go and be split. If this occurs, we have 2 pre-embryos. These will go and eventually develop, if they come to term, into identical twins. Now, if their connection is not fully split, it does not successfully split, those twins are going to be born conjoined. So, depending on where the division is, that's going to give us the full variety of conditions that can be associated with conjoinment. If two secondary oocytes are released from the ovary, fertilized by different sperm, then two pre-embryos may also develop, but these are going to be fraternal twins. So these two individuals are going to be genetically identical. They have all the same combination of DNA to work with. These two individuals, they each received genetic information from their mom, but it was two separate eggs, and they got two separate sperms from their dad. So they are just genetically brothers. So identical twins, a single fertilized egg split in early in development, making two pretty identical people physically. Fraternal twins, two oocytes split, were released and fertilized by two separate sperm. These two are genetically siblings. These two are genetically identical, and these two are also genetically identical. The split is incomplete, resulting in conjoined twins. So this is what happens when it does not split completely, caught at the head. It could be anywhere on the body. Now, cleavage. The blastocyst is going to be formed by day 6, and this is going to be a hollow ball of cells with a fluid-filled cavity in the center. There are going to be two parts, which is going to be the inner cell mass and a structure known as the trophoblast. So the inner cell mass is going to become the embryo proper. This is going to become most of the tissue of the actual baby. And some parts of it are going to become embryonic membranes that are not going to actually be passed on to the offspring. Trophoblasts give rise to the extra embryonic membrane. So this is going to be the embryo's contribution to the placenta. The rest of the placenta is going to be formed by the mother. Now, the placenta is a filtration organ. It's going to be delivering oxygen and nutrients to the embryo, and it's going to be carrying away CO2 and other waste to be disposed of in by the mother's body. So the actual implantation is going to be done by the blastocyst inside of the uterus after it's escaped the fallopian tube. So this is going to occur about 6 days after fertilization, and the blastocyst is going to become embedded in the endometrium. This normally occurs pretty high on the back wall of the uterus. Now, if it does manage to implant outside of the uterus, usually it implants too early, somewhere in the oviduct. This is known as an ectopic pregnancy, and without pretty extreme surgical intervention, it's a pregnancy that's going to go and like kill the involved parent. So these are pregnancies that are typically terminated. It's so early in development, there's not really like the baby never had any chance of developing really, and it's you can't just transplant it. So the only way to save the mother is to terminate the pregnancy at this point. Here you can see how that kind of works. So we have our ovum fertilized, our zygote going all the way down and then moving into the uterus. Now our blastocyst, it has developed enough to be a blastocyst. It's going to anchor itself to the endometrium. And then cells from the trophoblast, the parts that's going to become the extracellular and like placental organs, membranes. These are going to go and start to grow out and extend into the endometrius, which is in turn going to specialize and start forming the placenta. Now, implantation. So when the blastocyst anchors itself into the wall of the uterus, it's going to start releasing a hormone known as human chorionic gonadotrophin, also known as HCG. This hormone production is done by the blastocyst during early pregnancy. As pregnancy continues, it's going to be taken over by the placenta. What this hormone does is it maintains the corpus luteum in order to facilitate continuous progesterone production. So progesterone is the maintenance hormone for pregnancy. It prevents the shedding of the uterine wall. It prevents the induction of a reproductive cycle. It's going to go and keep them from menstruating, and so it's going to keep the pregnancy from terminating. This is the hormone that pregnancy tests are going to go and be scanning for. The one little stick test. This is what it's identifying. Now, the condition of infertility, it can happen to a male, can happen to a female, but it pretty much just means that if the medical definition of it is that if conception does not occur after a full year of unprotected sexual intercourse, They are having they have clinical infertility, and they might need to go and seek secondary treatment or secondary methodology. Oftentimes, it can be a very simple solution, something like a lump of scar tissue in the wrong place in the internal organs, or it could be a nutritional issue, it could be a stress issue or a medication issue. But the actual condition of being unable to reproduce for a year after trying is known as infertility. The implantation of the blastocyst is going to be one of the largest hurdles to overcome. In general, the further into development you get, the more sturdy the kid's going to be. So more can go wrong earlier. Now, embryonic membranes. You have four membranes forming around the preembryo. At the end of the pre-embryonic period, you have the amnion, the yolk sac, the allantois, and the chorion. Each of these are going to go and fulfill different portions. Or each of these are going to go and fulfill different functions, which we'll go into a little bit more detail. This is just them visualized. Here you have the embryo, and then you have your allantois, your yolk sac, your chorion vitae, all the things that we're going to be talking about. So the amnion, that's going to be this fluid-filled compartment that is going to fully enclose the embryo. It's serving as a protective cushion for shock absorption. That's its main purpose. Then you have the yolk sac. This is a site of early blood cell formation, and it's going to be the source of the primordial germ cells that are going to go and help facilitate growth of the embryo. So this is going to go and be like a mixture of stem cell. This is going to be a mixture of stem cells and nutrients and hormones that are all going to go and trigger and perpetuate growth. Then you have the allantois. This is the blood vessel that is going to connect the baby to the placenta. It's going to go and be a key component of the umbilical cord, which is a fibrous rope-like connection between the embryo and the placenta. So the allantois is the circulatory tissue of the umbilical cord. So we go back here, you can see the allantois right here, running back and forth, circulation. Now, the chorion is the outermost membrane, and this is going to be formed from the triploblast. This is going to be from the embryo's contribution to the placenta, and it's going to be mainly a respiratory organ. It's going to result in gas, nutrient, and waste exchange. This is going to be the interface between the baby and the rest of the placenta and the mom. Now, the placenta is going to transfer O2 and nutrients from maternal blood into embryonic blood. And it's going to take CO2 and urea from the embryonic blood into the maternal blood. That's then going to be filtered by the kidneys and by the lungs and expelled as waste. Now, it's also going to produce hormones essential for maintaining the pregnancy. It's going to produce very specific rates of HCG, estrogen, and progesterone. Progesterone is our pregnancy maintenance hormone. HCG is also going to go and work to prevent the start of the menstrual cycle. And estrogen is necessary for various stages of development, as we covered in the previous video. Now, forms from the chorion. Now, the placenta is going to be formed from a combination of the chorion of the embryo, again, the triploblast, and the endometrium of the mother. It's going to form structures known as chorionic villi, which are going to be finger-like projections to increase surface area that are going to then be able to exchange with the mother for nutrients, oxygen, and waste. Now, the placenta previa is where the placenta forms in the lower part of the uterus, and it actually will directly cover the cervix. This is going to interfere with the birth and often kill either mother or child. So, yeah, typically want to deliver by cesarean section if the pregnancy is brought to term. So, here you can see the different stages of development. The placenta begins to form about 2 weeks shortly after implantation and is fully developed by about 12 weeks. That's when all of the structures are fully developed. From there, it's just about growth. It's just about getting bigger. So, this is the Coriana villi. You see how it has these weird little finger-like projections to just increase the surface area with the mother circulation in order to go and try to make that exchange more efficient. Yeah. Interesting thing to note. You see how the veins and the blood vessels are like kind of spiraled around each other, especially at the base here in the umbilical cord. This is known as countercurrent circulation, and it is a way of conserving heat energy as we transfer blood between the two. Now, embryonic period. There are going to be three processes. Three processes produce the embryo. What? Oh, I skipped the slide. Okay, here we are. So, the embryonic period. First, it's going to go and start with gastrulation. And this is going to be the formation of the three primary germ layers, which are going to be the three layers of tissue that define everyone's body. We also are made out of these three germ layers as adult organisms. You have the endoderm, which is going to be the outer layer, the mesoderm, which is going to be the middling tissue, the tissue between the endoderm, and the ectoderm, which is going to go and be, oh, I'm sorry. Endoderm is the inner layer, like the lining of your GI tract or the lining of your lungs. Your mesoderm is going to be the tissue in the middle. And then your ectoderm is going to be your outer layer, so like your skin. At this point, though, these are just three layers of pretty undifferentiated tissues that are going to be forming a ball. So, so there are going to be three processes that are going to produce the embryo, and the embryo at this point is going to be defined as having a distinct humanoid appearance by the end of the embryonic period. So it needs to have all the human structures and be recognizable as a human being. So cell division, it's going to continue in the pre-embryonic period. This is going to go and be one of the foundational tools as one of the three processes. The first, cell division. Then cell differentiation. The cells start to become specialized with respect to structure and function. This is where you start to see the glory that is a multicellular animal. Before this point, none of these cells are specialized. They're all just cells. And then finally, you start to see morphogenesis, which is the development of overall body organization and shape. You're going to see the foundation of organ systems. You're going to see the foundation of like bones. You're going to go and see the basic form of the body. This is when you are visibly seeing like a little person forming. Now, gastrulation. Cells in the embryonic disc differentiate and migrate, forming the three primary germ layers from which all tissues and organs form. The ectoderm, the outermost, the mesoderm, the middle, and the endoderm, the innermost. Now, the embryo is going to be known as a gastrula at this point, and this is a very important part of morphogenesis. So here, if we go and look at this chart, we start with our morula. We remember the morula is just a ball of cells, complete solid all the way through. We anchor on the wall of the uterus, and then and then we see a blastocyst forming. So there's a hollow cavity. This tissue is going to form a trophoblast, and then there's an inner cell mass as well. During the later stages of the blastocyst, the preembryo within the embryonic disc, two layers of cell have started to go and divide the amniotic cavity from what is going to turn into the yolk sac. And then down here, you can go and see the start of the trophoblast as it's going to go and be preparing to go and anchor into the uterine wall. As time goes on, around day 16, we are now fully a gastrula, an embryo with three primary germ layers, the ectoderm, this outer pink one, the mesoderm, and the endoderm. So our endoderm is going to be the lining of digestive, urinary, and respiratory tracts. That's going to go and be like the outer edge of our, that's going to go and be the lining. That's going to be composed initially of the yolk sac. That's going to go and be the endoderm. Then we have the mesoderm, thin layers of muscle and bones that are going to start differentiating and emerging between these layers. Then you have the ectoderm, which is going to form the nervous system and the outer layers of the skin. So three different specialized types of tissue. And this outer layer is going to go and just turn into like this is going to go and turn into the extra embryonic tissues, like the nutrient providing layers and such, and shock absorption. Now, the ectoderm forms the nervous system, and it forms the epidermis and all its derivatives. That's going to be your hair, your nails, your oil glands, sweat glands, mammary glands. It also includes certain tissues in the brain, I like our and certain tissues throughout the nervous system in general. Or sorry, it's going to form certain glands that are associated with the nervous system. In general, if it is designed to emit a lot of hormones, it's going to go and be made out of glands, and therefore it's going to be made out of ectoderms. And then we have the mesoderm. This gives rise to muscular, bone, connective tissue, and organs such as the heart, kidneys, ovaries, and testes. This is also going to form the notochord, which is going to go and be the precursor to our spinal cord. Now, the notochord, flexible rod, and it's going to define the long axis of the embryo. It's going to run along the length of what will be the spine. It's going to go and give it some rigidity and some structure, but it's also going to induce neurulation. So nervous tissue is going to start saturating that area and forming nervous structures. This is going to go and help us form our spinal cord. Now, the endoderm is going to form the lining of the urinary, respiratory, and digestive tracts. It's also going to form some organs and glands. So it's going to be your pancreas, your liver, your thyroid, and your parathyroid glands. Also, this is tissue, basically tissue that is designed to have a quick turnover. So if the tissue needs to be replaced very frequently, it's probably going to go and be endodermic tissue. Now, neurulation is going to be the development of the central nervous system. The embryo is known as a neurula, and this is the point where we're going to start forming the neural plate. So this is when the notochord induces the overlying ectoderm to thicken and start to specialize into nervous tissue. So this is how we're going to go and start forming the neural cord from neural plates. Now, as the neural plates are forming, depression is going to form in the center of them, which is known as the neural groove. Yeah. So as the groove deepens, it's going to go and saturate with fluid. And from one end, the brain is going until it forms like a tube that is just filled completely with fluid. Eventually, it seals over again at the top. So it's like a hollow tube filled with juice. Then the brain is going to start developing from one end of it. And then the spinal cord is going to start growing from the other end. Now, a birth defect of the neural tube is known as spinal bifida. This is where the part of the spinal cord develops abnormally. Some can be corrected with surgery, but very often it's just going to manifest as partial paralysis. Then there's anencephaly, which is incomplete development of the brain. This leads to stillbirth or death shortly after birth. Can't do a lot about it. So right here, this is an example of spinal bifida. So the baby's spinal column at the base of its back did not develop properly and made all this other exothermic tissue. So luckily, because of how low in the back it is likely that the baby will not be paralyzed. And if it is paralyzed by this, it most likely will still survive the procedure. But yeah, it can be pretty rough. So here at the top, you can see ectoderm overlying the notochord thickens to form the neural plate, just a flat plate of tissue. The neural plate starts to fold over on itself until it makes this neural tube, which is going to be filled with spinal fluid. It's going to go and fold and then start to curve. And this is going to fill up with spinal fluid. Now, from there, these dark cells, these actually are going to form the peripheral nervous system. So all these pale blue cells, these are part of the central nervous system. This is going to form your spinal cord, your brain, all that good stuff. But this dark blue, that's going to differentiate and innervate until it makes the axial nervous system. So that the peripheral nervous system or axial nervous system is going to saturate the limbs as well as certain aspects of the torso and the face. This is and this is the point when it's going to start differentiating away from central nervous tissue. Now, at this point, somites, which are going to be formed by the mesoderm, are going to develop on either side of the neural tube. These are going to go and eventually develop into the muscular tissue and the bone tissue that's going to go and form your spinal column and all the supportive musculature around it. Now, somites are going to be blocks of mesoderm, we just talked about them, that are going to form alongside the neural tube. They're going to go and take the form of skeletal muscles of the neck and trunk, connective tissues, and vertebra. So, again, all the structures that are not actually part of your nervous system here. Now, develop in the reproductive system. So gender is going, or sex is going to be determined. Gender is a sociological construct, not a scientific construct. So we do not acknowledge it. Sex is going to, well, not acknowledge it. I mean, like it's important. It's important for people to, it's important for people to have identities. At the end of the day, just don't fucking pick on people. It's mean. Be nice to people. Anyway, so every female is going to go and have two X chromosomes. Every male is going to have an X chromosome and a Y chromosome for genetically healthy individuals. Because of this, sex is going to be determined by the male because either a Y sperm or an X sperm would be able to fertilize the female egg and go and contribute that genetic information. If you end up XY, you end up male, but you can only get Y from the male. So sex is determined by the male. The female has XX, which means that she doesn't get to determine the gender of the baby, but she does have two copies of the X chromosome, which can be super, super useful if you have a recessive sex-linked genetic disease. If you have a recessive sex-linked genetic disease, then you're going to go and see if you have a normal chromosome, like let's say you have one X that's infected with the bad gene, but you also have a normal chromosome. Well, the normal chromosome is going to overpower the phenotypic expression of the diseased sex-linked chromosome. If you're a male and you have a sex-linked recessive disease on the X chromosome, you do not have any other X chromosome to overwrite that. So instead, it's just going to be fully expressed. Now, development of the testes in an XY embryo. So that's going to start about 6 weeks after fertilization. It's going to go and be mainly modifying pre-existing female structures. So development of the testes and begin about 6 weeks after fertilization. It's going to be prompted by the SRY region on the Y chromosome. Now, testosterone is going to be the main hormone that's going to go and be inducing these physiological changes. Testosterone is going to be produced by the testes. And it's going to go and then direct the development of the male reproductive organs as well as the secondary sexual characteristics associated with masculinity. Now, female embryos lack a Y chromosome. Without the presence of the SRY gene to tell the ovaries to descend and start specializing into the structures that would go and turn into testicles, they stay where they are and they continue specializing into female reproductive organs. So, femaleness is defined. So, yeah, maleness is coded for on the Y chromosome, and it's going to be using female structures and then modifying them for use. Now, because of this, during formation, you can go and actually see how the male structures are going to be emerging out of female structures. So here you have a clitoris, labia majora and minora. This is all going to be coming from these foundational structures. So there you got the butt. You have the labial scrotal swelling. This is either going to become a labia or a scrotum. You have the urogenital groove. That is the separation line in the scrotum that in a female is going to split to become the entrance to the reproductive tract. And then this bud is going to either become a clitoris or become the head of a penis, depending on whether testosterone is present or not. Here you can see how the two tissues are differentiating over the course of 10 and 14 weeks and how they're specializing into their final structures, but derived from the same initial tissues. Now, here's a little video that can go and talk about the prenatal period to a certain degree. So let's have a look.
Reproduction. Ovulation provides the opportunity for pregnancy. The ovary releases the egg into the oviduct, where it may fuse with a sperm cell, a process called fertilization. A fertilized egg is a zygote. About 1 day after fertilization, the zygote undergoes a rapid series of mitotic cell divisions called cleavage, first resulting in 2 cells, then 4 cells, then 8 cells, and so on, until by day 4, the solid ball of 12 or more cells is called a morula. The morula is no larger than the original fertilized egg. As cell division continues, a cavity forms at the morula's center, converting the morula to a blastocyst. The blastocyst has 2 parts, an inner cell mass, which is a group of cells that will become the embryo proper, and a trophoblast, which is a thin layer of cells that will give rise to part of the placenta. The blastocyst attaches to the uterine lining called the endometrium and digests its way inward. The blastocyst increases in size as it continues to develop. In the late blastocyst stage, the inner cell mass moves away from the surface of the blastocyst and becomes a flattened, plate-like structure called the embryonic disc. At the same time, an amniotic cavity forms, filled with amniotic fluid. The cells within the embryonic disc begin to differentiate and migrate, forming 3 primary germ layers known as ectoderm, mesoderm, and endoderm. The cell movements that establish the primary germ layers are called gastrulation, and the embryo during this period is called a gastrula. Ectoderm will eventually form the outer layer of skin and the nervous system. Mesoderm will form muscle, bone, and other connective tissue, and various organs, including the heart, kidneys, ovaries, and testes. Endoderm will form the lining of the digestive, urinary, and respiratory tracts, and a number of other organs and glands. Gastrulation paves the way for the development of the nervous system and other organs. A flexible rod of mesoderm tissue called the notochord develops where the vertebral column will eventually form. At left is a dorsal view of the embryo. Both views depict day 17 of development. The notochord prompts the overlying ectoderm to thicken and form a nervous system precursor called the neural plate. The neural plate folds inward, forming the neural groove. The neural folds grow upward and fuse, forming the neural tube, which will ultimately develop into the brain and spinal cord. The process of neural tube formation is called neurulation. The formation of the central nervous system from ectoderm is a major milestone in embryonic development.
Alrighty, yeah. Sorry, to be honest, I have a bit of a stutter, and my nerves definitely get to me sometimes. So I appreciate having these little videos in there. If I have a model, I'm having difficulty conveying, and I hope you guys appreciate it too. So the fetal period is going to continue growth and differentiation of tissues and organs, which is going to increase the fetus length as well as the weight of the animal of the creature. Remember, this is a fully formed animal, fully formed human, whatever, and it has all the structures. They just need to get bigger at this point. So this is the beginning of what we call the allometric growth period. So there's going to be differences in growth rates of in various parts of the body, which is also going to change the proportions of the animal over the course of its life. So this occurs both before and after birth, and this is following the course of like a human female over the course of their life. So as you can see, as they age, the head is going to get smaller proportionally until we enter adulthood around here. And then we're going to have a slight loss in height and stuff. This is also covering other changes associated with aging, obviously. But the main key point here is going to be it's going to be the progression of the tissues growing at different rates in order to go and create a more proportionate adult animal. Now, fetal circulation, the lungs, the kidneys, and the liver. These do not perform their postnatal functions in the fetus. Blood is being filtered mainly by it's going to be filtered mainly by the placenta. And in general, it's going to be shunted past these organs through temporary blood vessels or openings. Oh, one of these, the first of these shunts is going to be known as the ductus venosus, and this is going to go and allow most blood to bypass the fetal liver, and go directly towards the inferior vena cava. Then we also have the foramen ovale, which is a small hole in the atrial wall between the right and left atrium, and this is going to allow blood to bypass the lungs. This hole sometimes does not reseal. This is one of the most common heart problems that people tend to get. Like if someone has a heart murmur, it's typically because this hole did not seal. Now, ductus arteriosus. This is going to connect the pulmonary trunk to the aortic artery. This is going to divert most blood away from the lungs. So all these are temporary shunts that are not going to be used after the birth of the baby. So at birth, all organs take on their postnatal functions. Fetal circulation converts to the postnatal pattern. The ductus venosus and the ductus arteriosus are going to constrict, shrivel, and then go and be used as the foundation of ligaments later. The foramen ovale closes. If it does not close, this is going to go and result in a blue baby, and require pretty immediate surgical intervention to keep the infant alive. I say animal. My background's veterinary, so that's what I'm thinking. Now, if we look here, you can go and see these two babies. This one it's foramen ovale on the left. It's foramen ovale. Failed to close. And as a result, we are having blood leakage between the two sides of the heart. This also is what it's going to look like inside of the baby. This is what it's going to look like inside of the womb. When this is an undeveloped baby, these shunts are going to be working, and that's going to be a healthy function. After birth of the baby, those shunts should be closed to go and restore the four-chambered closed circulatory system. We do not want an open circulatory system. Yeah. Yeah, and here you can see the umbilical arteries become ligaments right here, that flow of circulation, and this is where, and the belly button is going to be the point where the placenta actually connects to the infant. Now, parturition. So, 38 weeks after fertilization, this is where you transition from prenatal to postnatal development. It's time to give birth. So, this is going to go and be working through a positive feedback cycle. As this hormone is going to trigger a behavior, and that behavior is going to induce more release of that hormone. It's going to encourage itself to keep going. This is a positive feedback cycle. So oxytocin from the posterior pituitary gland prompts uterine contractions. Labor, the process by which this induces labor. So labor is going to go, and this is the process by which the baby is born, by which the fetus is expelled from the uterus. It's going to go through 3 stages. These are dilation, expulsion, and the placental stage. So dilation is going to begin with the onset of contractions. It's going to continue until the cervix is fully dilated. This is the longest stage. When someone's water breaks, this is what is happening. Then we have the expulsion stage. So this is when the contractions are becoming more and more frequent. This is when the baby is actually going to be entering the vaginal canal, and this is going to end with delivery of the baby. After the baby is expelled, we begin the placental stage. So after the baby's out, the placenta has to leave too. It's a lot of extra tissue. This is something that you also need to go and reassure, because otherwise it can calcify and kill the mother. So here you see dilation stage, the cervix starts to go and dilate. It can dilate up to 4 inches. Then expulsion. So the baby's going to start to slide out into this. Muscular contractions are going to be forcing the baby further and further and further. Oxytocin is going to be inducing these contractions. The baby's presence in the vagina is going to induce more oxytocin, more contractions, more hormones, just get stronger and stronger until the baby pops out. Then we have the placental stage after the baby's been born. This is going to be like a partial shedding of the uterine wall. and it's going to go and release this tissue, which then can be expelled as one large mass. Now, the positioning of babies during birth is very, very important. So you can't really deliver a baby buttocks first or backwards in any species. Without pretty intensive intervention, breech births tend to be lethal. Now, the baby can usually the baby's going to be born head first, facing the vertebral column of their mother, so face out. And this is going to be good. This is going to be a standard delivery. Breech birth is very bad. Breech birth typically is lethal. You often require surgical intervention. The way that you typically end up having to correct this is you try to turn the animal or the infant around inside of the womb. You do this with cattle and horses pretty frequently. Now, state of development at birth, full term infant. This is a baby born at least 38 weeks after fertilization. Premature, it says the baby is born before 37 weeks of gestation. So if you were a preemie, you did not make it to the full 38 weeks. Now, developmental defects present at birth, they may concern structure, function, behavior, or metabolism. So let's start with genetic developmental defects. This is going to be issue with the number or structure of the chromosome. So damage to the genetic information that's going to be coding the actual creation of the organism. This can be induced by environmental factors, anything that can go and result in genetic damage or induction of cancer. So that's going to be like things like drugs, radiation. It can even just be standard. It can even just be inherited genetic issues, from a parent. So because of this, most of these are going to go and be, if it's going to be terminal, it tends to be during the pre-embryonic period. If there's a genetic error, there's a lot of like stops that are going to occur during different stages of the pregnancy to ensure that the baby is like developing the way it should. And if the body does not believe that it is, it might terminate the pregnancy. So here, pre-embryonic period, cleavage and implantation, 1 to 2 weeks. Exposure to a harmful agent during this may cause spontaneous abortion. And then from week like 3 up to 8, we're in the embryonic stage. This is the exposure during this stage could lead to extremely severe birth defects. General, the further in you go, the less severe the impact is to the baby, simply because there's more baby to handle like filtration and things like that. The closer you get to birth, the less damage is going to be done by exposure to this stuff. But as you can see, up until like throughout most of the pregnancy, the central nervous system, the ears, and the ears are going to go and be subject to they're going to be subject to extreme birth defects. And if you have something wrong with the central nervous system, even minor defects are extremely noticeable. Like if you have slight brain damage, you're not going to shrug that off. Now, moving on from that, let's talk about lactation. So lactation is going to be the production and ejection of milk from the mammary glands. They theorize this may protect against breast cancer, simply because it's going to go and be removing tissue. But I have not, since that claim was made roughly 8 or 9 years ago, I have not heard anything else about it. So I'm not inclined to believe it. As you can see, the copyright for this presentation is 2017. So now lactation. So lactation is going to be induced by a couple different hormones. The main two are going to be prolactin and oxytocin. So prolactin comes from the anterior pituitary, and it's going to go and produce milk. It's going to stimulate milk production. Oxytocin is going to come from the posterior pituitary, and it's going to stimulate milk release. Now, when a baby is born, it does not have a well-developed immune system. In general, the hope is that it's going to go and get acquired immunity, which is when part of the immune system of the mother is temporarily donated to the baby while its organs are getting up to snuff and it's figuring out how to have an immune system. So, in order to go and pass the immune system from the mother to the baby, we give them what is known. The mother immediately breastfeeds after birth, and that milk is going to be temporarily saturated with antibodies, big important component of the immune system. Those antibodies are going to serve as the baby's immune system for a couple days while its organs kind of get the way that they should be acting. After about day 3, the mother's not going to be producing colostrum anymore. But that's why in the first 2 or 3 days, nursing is super, super important. It's the way that we're going to be building the baby's foundational immune system. Now, hmm, colostrum and milk both are going to contain a certain number of antibodies, and they're also going to contain specialized proteins that are going to facilitate boosting the immune system. But colostrum is the dedicated immune fluid. Now, period of growth and developments after birth. So stages from birth to 12 months, we call that infancy. Childhood, 13 months to 12 or 13 years. Adolescence, puberty to late teens. And then adulthood. Biologically, you finish developing at 25. In general, most of the important systems are in place by about 21. So, that's why we let 17-year-olds take out student loans, because it's easier to trick them into it. Now, aging, normal, progressive, everyone ages. You're all going to die someday. That's okay. That is just the steady decline in the structure and functions of the body of adults. And it's going to go and lead to slow changes in the organ system over time. That's kind of the other side of this. You have growth up until you reach your adulthood, and then at the end, you're going to have decline in function just due to deterioration. That's normal. It's fine. Here are all the organ systems, a bunch of organ systems and changes that occur with aging. Uh, it's important to go and note again that the reproductive changes are going to be while they are the most extreme in females because of menopause, males also have some pretty extreme changes to their system. The prostate gland enlarges, which causes painful and frequent urination. You're going to go and see erectile dysfunction. You're going to go and see lower quality sperm with higher genetic issues. There's a lot that happens. Now, possible causes of aging. So, aging in theory is your body breaking down. It is not your body working as it is intended. So, that's going to be changes in the critical organ systems, secession of cellular division, damage to the DNA and other macromolecules. We think a big part of this has to relate to a structure known as a telomer, which is going to go and be protecting the outer edge of DNA. As you age, that breaks down, and then your actual DNA starts to get broken down instead. But yeah, normal biological process cannot be slowed, stopped, or reversed. Most anti-aging treatments are a rip-off, so don't spend money on them. Life expectancy for babies born in the U.S. In 2017, it was 79 years. Now it's closer to 73 years due to economic factors mainly. More people are poor, more people are stressed out. So yeah, that's actually something that is worth discussing. It's been less than 10 years, and we've lost almost 10 years off our life expectancy in the last 10 years. That's pretty fucking bad. Lifestyles, factors over which we have personal control. These are things that you can actually change to try to get closer to your maximal age, live past the life expectancy. Proper nutrition, a lot of exercise and sleep. Avoiding drugs. You don't smoke, don't take weed, don't take coke, all that stuff. And of course, routine medical checkups. If they catch the stuff early, you usually can go and do something about it. Now, at this point, we should be able to differentiate between prenatal and postnatal periods of development. Should be able to use correctly the following terms to describe the prenatal development: fertilization by one sperm, zygote, cleavage, morula, blastocyst, inner cell mass, trophoblast, placenta, implantation, gastrulation, germ layers, and neural tube development. Should be able to list and order the stages of labor and describe the birthing process. And you should be able to relate the prenatal developmental timeline to the seriousness of a birth defect and describe potential causes of different birth defects. You should be able to explain control over milk production and letdown. And you should be able to describe postnatal growth and development and the factors that influence aging and high quality of life through old age. All right, folks, know that was a lot to throw at you. Again, I'm going to be making I'm going to go and be reworking some of our materials, and I'm going to be pulling from the presentations. In theory, if everything goes well and God is good and I have enough unpaid time to completely rewrite this course they gave me, because that's how this works. They give you an entire course, they give you a course that is completely made, and then they say you're allowed to teach whatever you want, change it however you want. But they don't pay you to do that. They don't give you any time to do that. So this class needs another 400 hours on it, and I'm not being paid to do that. So we're trying to figure that out, but I am making small incremental changes. The biggest one that I'm making is I'm completely rewriting every exam, every quiz. I got some feedback from students. They really didn't like the formatting of them. The material, the professor who designed them initially, quite frankly, they didn't do a good job. So I'll be doing what I can as fast as I can, and I have been. With that, I wish you all good luck. Have a great day. Bye bye.